Well, hello and welcome to the Veterans Day video on the structure of DNA. It's actually 9 o'clock on Tuesday, but I promised you I'd make the video, and here it is. So sort of after we discussed all the experiments, Hershey and Chase, Avery, which sort of biologists or scientists came to the conclusion that DNA and not protein was the genetic material. So if you remember, we discussed that most scientists before the Avery and Hershey and Chase experiment believed that it was protein that was the genetic material. But after the experiments of Avery and Hershey, scientists were pretty convinced that it was DNA that was the genetic material. The sort of next sort of challenge that scientists had was to figure out what is the structure of this DNA. They sort of knew the components, but they didn't know how the components were organized or structured. So the structure of DNA was sort of determined, or used to determine the structure, was a tool known as X-ray crystallography. The first sort of person to use this technology was Rosalind Franklin, which is back in this time was sort of one of the few females in science. And essentially an X-ray crystallography, we don't have to know the details, but so this here would be sort of your DNA that you extracted. And they send an X-ray beam, and you don't have this slide, I added it, but you don't need to know all the details, just sort of general of what happens. They send an X-ray beam through the crystallized DNA structure, and then the rays diffract, and using the diffraction pattern you can get an idea of the structure of the molecule. Now, interesting sort of side story, Rosalind did a lot of this work, but her pictures that she took were stolen by Watson and Crick, and there's actually a movie about this that we watched in college. And they were the ones that get credit for discovering the structure of DNA. They stole her pictures, and then she died. And so even though most of the work, or most of the pictures that Watson and Crick used were from Miss Rosalind Franklin, Watson and Crick got most of the credit for discovering the structure of DNA, when really Rosalind did most of the work. So by looking at these pictures that they got through X-ray crystallography, they deduced that the structure of DNA was a double helix. And it's sort of two strands of DNA that are held together by hydrogen bonds. We're going to sort of look at this much more closely on the next slide. So there's sort of a lot in this slide here that we're going to take a look at. So from the pictures, and my pen is not wanting to work. So from the pictures, they were able to conclude a lot about the structure of DNA. First, that in general, it was that the structure of DNA is composed of two anti-parallel, we're going to discuss what that means, sugar phosphate backbones. We're going to take a look at this on the next slide, with nitrogen bases bonded together through hydrogen bonds in the interior of the molecule, or the inside of the molecule, and we're going to take a look at this. And it was also, from the pictures, thought that A pairs with T and G pairs with C. So if we take a look at this picture, there's sort of a lot to see here. So first we have a nucleotide, which we learned about in the first unit. Here we have a phosphate right here. Then this is the sugar deoxyribose. And then these are the nitrogen bases. And you see that they're connected to each other. The sugar is connected to the phosphate is connected to the sugar. The phosphate is connected to the sugar, and so forth. This here is called the phosphate-sugar backbone. And it occurs on this side as well. Phosphate sugar backbone. And in this molecule there are 1, 2, 3, 4, 5, 6, 7, 8 nucleotides. But this is a strand of nucleotides here, and then this is a strand of nucleotides here. These strands are held together in the middle by hydrogen bonds. The rules state, and we'll talk about why it has to be this way, that A can only pair with T, and G can only pair with C. Now, sort of the big question here is, why is this structure said to be anti-parallel? Okay, well, if you take a look on this strand here, okay, this strand has the phosphate group facing up. Okay, and this strand here has the phosphate group facing down and the sugar end facing up. Because of that, the molecule is said to be anti-parallel. This side is said to run in a 5', because this is the 5' carbon. We'll talk about this more on Thursday. This is the 5' carbon of the sugar. To the 3', this is the 3' carbon direction. So the slide, or the strand that has the phosphate group up runs in a 5' to 3' direction. And the strand that has the phosphate group down runs in a 3' to 5' direction. More about this on Thursday. But what it means to be anti-parallel is that the strands are flipped relative to each other. This one has the phosphate group facing up, and this strand has the phosphate group facing down. So Watson and Crick saw that there was this sort of specificity in pairing, that if we look back here, that when the strands are held together, A has 2 hydrogen bond with T, and G has 2 hydrogen bond with C. And each pair actually forms a different number of hydrogen bonds. When A bonds with T, it forms 2 hydrogen bonds. And when G bonds with C, it forms 3 hydrogen bonds. That actually makes a G-C base pair harder to break, or a G-C base pair is stronger than an AT base pair. So if you have a strand of DNA molecule, or a DNA molecule that's composed of a lot of GC base pairs, it's harder to break apart than an AT base pair would be. So this is sort of showing the hydrogen bonding. And if you remember, when you have a hydrogen bond, you have to have hydrogen bonded to a very electronegative element. And the very electronegative elements are F, O, and N, but F doesn't really do biology. And this H is going to hydrogen bond to another very electronegative element. So those are sort of the elements that you need to have a hydrogen bond. And these hydrogen bonds sort of fit together. The nitrogen bases have the correct chemical groups so that they can align and hydrogen bond. This one can only form 2 hydrogen bonds, you see, because this O could hydrogen bond, but there's not an H on this side to hydrogen bond with it. So it can only form 2 hydrogen bonds. Whereas you sort of see over here, this O on the C has a H to hydrogen bond with over here, which is not present here, which is why a G can't bond, C, that's why these pairs have to fit together. O can't bond with this one, the hydrogen bonding pairs don't work there. And sort of this one can't bond with this one because this OH here, this O can't hydrogen bond with this O over here, so it just doesn't work. Like the shape of the bases allows them, or the chemical groups of the bases allows them to hydrogen bond together. You'll also notice that A, which has 2 rings, always hydrogen bonds with T, which has 1 ring, and G, which has 2 rings, always hydrogen bonds with C, that has 1 ring. So sort of a 2-ring base, nitrogen base, always hydrogen bonds with a 1-ring hydrogen base. But I mean, the point is that these can hydrogen bond to each other because the groups line up so that a hydrogen bond between a very electronegative element and a hydrogen next to a very electronegative element, these groups are next to each other in an AT, when AT and put together, so they can hydrogen bond with each other. Okay, so this is what we did. I think this lesson that we did right now took like a week in regular biology. So this is a strand, and I'm going to struggle with writing without. I'm actually home, so I'm struggle with writing the letters without smartboard marker because I'm doing it with mouse. So this is a strand of DNA, and that usually they just show the nitrogen bases, but you have to remember that attached to each of these is a sugar and phosphate, and they're bonded together by a sugar phosphate backbone. So what's not shown here, sort of like how these molecules are connected, is a sugar phosphate backbone. And a lot of times they ask you, or you should know, if this is the one strand, what would be the complementary strand, or the strand that would be hydrogen bond to this strand? So one of the things to remember is that the strands are anti-parallel. And this strand is running from a 5' to 3' direction. That means in this strand, the phosphate group is facing up. So the complementary strand has to run in a 3' to 5' direction, and the phosphate group will be facing down. And then you sort of have to remember what bonds with what. So A bonds with T, T bonds with A, G bonds with C, C bonds with G, and then A bonds with T, and so forth, and so forth. I'll finish it out. My letters are horrible, but welcome to Writing with a Mouse. C, G, A. And if you remember, just to sort of represent, a AT base pair has 2 hydrogen bonds, so we'll draw 2 hydrogen, 2 dots, and a GC base pair has 3 hydrogen bonds. Just to sort of make sure that we know this, so ATs are easier to break apart or than GC base pairs. So 3 hydrogen bonds, and so forth, and so forth. Tomorrow, or I guess depending on when you're watching this video, I'm going to talk about how DNA is replicated. And the fact that that DNA is anti-parallel, this sort of idea is really important, that one strand runs in a 5' to 3' direction with the phosphate group up, and the other strand runs in the 3' to 5' direction with the phosphate group down, has consequences for DNA replication. And that's sort of going to be our discussion, DNA replication. Some of you may remember DNA helicase and DNA polymerase, and DNA ligase we might have talked a little about in 10th grade. But the fact, we didn't go into this detail, but the fact that the structure of DNA is anti-parallel causes problems for replication, and we're going to talk about how those are fixed. The last thing I want to make sure that I sort of focused on here, because I might have overlooked it, as even though we show DNA here as a ladder, in actuality it twists to form a double helix structure. Here we sort of showed it as a ladder to sort of look here at the hydrogen bonds in more detail. But in actuality, DNA forms a double helix and sort of twists. And if you ever take sort of advanced molecular biology, you sort of learn that this is called the major groove and this is called the minor groove. And those have different roles in sort of a DNA structure. But this is actually the shortest video yet, only 12 minutes. But answer the guided questions, and remember you also have to make the corrections on your photosynthesis and respiration tests for homework and upload it to the Dropbox. Enjoy your Veterans Day off, and I'll see you guys on Thursday.